Okay, so good afternoon, everyone. My name is Reese. Unfortunately, I can't be with you in person at the poster presentations as I'm currently overseas in Europe. However, I thought it would be good just to do a quick little presentation, a little bit of a talk regarding my project, which was looking at creating compounds using solid phase synthesis for use as radiopharmaceuticals to treat melanomas. So a little bit about, I guess, what we were trying to do. The main aims of the project were to use solid phase synthesis to try and produce this compound, which we're calling compound A for simplicity. But effectively, we were producing this peptide backbone, trying to attach an organic linker to it, and then attaching or creating a chelator to hopefully then bind to a radioactive lead particle. And how the chelator operates is it uses these donor atoms, the electrons on those donor atoms, to bind the lead into a stable coordination geometry so that it would carry it to the active site, to the receptor of interest that we're trying to target. And the reason that this compound was chosen was because it has a suitable pharmacophore or a special sequence within this peptide chain, which would mimic other molecules that would bind to the receptors that we were looking at. So this is sort of another schematic, just sort of explaining what we were trying to do. We're really creating this compound to drag or to bring the radioactive compound to the cancer cells is really essential, really, for this targeted alpha therapy, because the radioactive compound is only going to be effective on short ranges, roughly only about 100 micrometers, which is really only a few cell lengths in distance. So it needs to be in close proximity for it to be effective. It also means that we can minimize the off-target effects so that the radioactive compound isn't just damaging healthy cells because it's not moving around the system in an uncontrolled way. We're really having full control as to where it needs to be and how it's going to get there. We also decided lead was chosen as a very effective alpha emitter because it follows this process of decay to stable lead, which can still be chelated out of the system at the end, but also the half-life is relatively short. So it loses potency after it's reached its target, and it's not going to remain radioactive for an extended period of time. And really, this is just showing that melanocortin-1 receptors that we were trying to target, compound A can mimic alpha MSH, which is alpha melanocortin stimulating hormone. It mimics the binding of this with that pharmacophore. However, it doesn't have a cis-R bond like alpha MSH does, meaning that once it binds to MCR1, it won't be degraded as easily. It doesn't readily degrade as a signal, meaning that it'll stay there and allow the lead to radiate the cell. It's also worth noting in this process that MC1R receptors are overexpressed in melanocytes due to the fact that they regulate the production of melanin. And we commonly see in melanomas that the production of melanin is overexpressed, meaning there's an excess of melanin, which causes the production of moles and other visual signs of melanomas. So it's quite common to see this overexpression of MCR1 receptors, and this is why we chose it. This is why it's a good target for this sort of therapy, because there's a strong comparison between the overexpression in cancer cells and the normal expression in normal melanoma, in normal melanocytes. So in order to produce the compound A, we tried using solid phase synthesis, also known as SPS. It's a very effective tool for organic chemistry because it's a very quick and easy way of reducing peptide chains by adding amino acids from the C terminus onwards. And it's quite effective because all you need to do is add the amino acids in sequence and just wash and deprotect them as needed. So they've got protecting groups that prevent any side chains from forming or any unwanted reactions. but effectively you can do a series of different amino acid additions in a relatively short amount of time just by adding to the resin in sequence and then cleaving it at the end. So we did this and then we ran it through HPLC to see if we had produced compound A of the compound of interest. We were expecting to see a peak at around 15 minutes based on the information and previous synthesis that were performed. However, we saw this peak at around 13 minutes, which indicates that we did produce some protein of interest. However, it may not be the protein that we were looking for in terms of it being compound A. Nevertheless, it was indicative that the SPS was to some extent successful. We did then run it through mass spectrometry to see whether or not any of the peaks that we were looking for would be present. We using digital libraries, we had an idea as to what the fragmentation patterns would be and what the fingerprint sort of peaks we would see. We were expecting to see a fragment at 1476 mass to charge ratio with a +1. Unfortunately, we did not see that, indicating that whatever protein was there, it wasn't compound A. And we had no real way of sort of determining the identity of those fragments. And it was also very, it sort of suggested that whatever samples we did have weren't pure. And it was very difficult to differentiate between the background information and the peaks that we did have. So whilst not, I guess, fulfilling the aims of the experiment, we were able to, I guess, show that SPS has merit as an organic tool. And certainly it can be used in that sense if we were to replicate this information and try and produce more compounds. So that's it. Hopefully you've learned something. Hopefully you enjoy the rest of the posters and the presentations that are on offer tonight. I do regret that I can't be there with you in person, but I do thank you for listening and all the best.